Hello everyone, and welcome to this, our first video, in which I'll be showing you how to get the sequence for your gene and its promoter. But before we talk about sequences, it's important to talk about the structure of the gene as well, because a gene is not simply a bunch of codons back to back. Instead, there's a lot of complexity in a given gene that is very important to understand before you try to mutate it with Cas9, for example. So first of all, we can break a gene down into exons and introns. The difference there being that exons in a gene are going to be retained in a mature transcript, whereas the introns are going to be cut out after transcription. So you can see here this intron does get transcribed, but it is cut out at some point before this transcript reaches the cytoplasm. So if I'm trying to mutate a gene, for example, I would not want to mutate it within the intron because that's going to have almost no effect on the translation of the target protein. Instead, I would want to put my mutations in these exons here. But at the same time, it's also important to mention that a given gene might have a transcript that's much bigger than its given coding sequence, or CDS, as shown here. So for example, the start codon for a gene might be well into the transcript, giving us what we call this 5' untranslated region. Likewise, on the other end, after the stop codon is what we call the 3' untranslated region. These are sequences that get copied and retained into the mature transcript, but they do not contribute to the translation of the final protein. So likewise, you also would not want to mutate the gene in one of these UTRs because it would likely have very little effect on the final protein. We can get all of this information about the gene from this database right here called NCBI Entree. So I'll show you how to use that in a few minutes here. But overall, you can see that it's very important that we know these things about the gene before we start to design mutations or gRNAs to be used with Cas9. In addition, upstream of the gene is what we call the promoter. So the promoter region is going to have a few important aspects to it. First of all, it's probably going to have what we call a TATA box, shown down here as TATAWAW. Those W's could be A's or T's, just FYI. And that TATA box is going to be one of the main things that recruits RNA polymerase, the enzyme that transcribes our gene. So this promoter region up here is very, very important because it enables RNA polymerase to bind. And in addition to RNA polymerase, it might also bind other what we call transcription factor proteins that also bind specifically to these sequences. And they can either, let's say, block RNA polymerase from binding to repress a gene, or they can actively bring in RNA polymerase. They can encourage it binding to increase the transcription of a given gene. So the promoter is in many ways just as important as the gene sequence itself, because if you don't have a promoter that is active, that is recruiting a bunch of transcription factors, recruiting RNA polymerase, then you won't see any expression of your gene. So both of these regions are very important. And I'll show you how to use these two tools, EPD, the Eukaryotic Promoter Database, to look up the promoter sequence. And then finally, we'll also use this promo tool here to search that sequence for putative transcription factor binding sites. Altogether, this should give you a very clear understanding of what your gene is and how it's regulated. Okay, so let's start off by getting the sequence of your gene itself. For that, we're going to go to this website right here, this NCBI Entree. I'm going to click on that hyperlink. That will bring up the web page here. And what I want to do first is type in my gene name. So for example, we're going to be using interferon lambda 1 in this exercise. I also want to type in my organism name. So I am interested in the human version of IFN1. All right, so then I'm going to click search. And this is what you should see first. An entry looks something like this. Now, it's worth mentioning that there are a lot of entries for any given gene in this database, but we want the one that has been curated, the one that most of us agree is the correct entry, because some of the entries in this database are actually outdated. They contain errors that have recently been fixed. So the best thing you can do is come down here and click on this plus icon, and it'll give you all of what we call the RefSeq sequences. These are ones that are trusted reference sequences, and the RefSeq select entry is going to be the best one you can find. It should start off with an NM underscore and then some numbers. So let's click on this one. This looks like the best choice for IFNL1. And here's all the information we have for this transcript. Now notice here, this is not the sequence of the gene itself that would exist in the genomic DNA. Instead, this is the transcript of that gene, and it's the processed mature transcript. So all we're going to see here will be exons. But anyways, that being said, we've got the gene identifier here, IFNL1, the organism, Homo sapiens, and the accession number. So you want to save this accession number somewhere. You want to write that down so that you can come back to this exact entry later, just in case you need to. Again, NM means it's a RefSeq, and it's a RefSeq for an mRNA sequence. That's what the M is for there. And you'll notice it says 0.2 here. This means this has been revised twice. All right, and then we can scroll down. There's a bunch of papers where this sequence has been referenced or where it came from. Keep scrolling. Short description of the gene there. And then down here is some very important information. So first of all, you have the gene entry. What this is saying is that you have a transcript that is 775 bases long. Now within that 775 bases, you're going to have exons. This first exon spans from 1 to 186. The second exon, if we scroll down here, spans from 187 to 264. So, like I said before, there are no introns in this sequence. The exons are going to be directly against one another, 186 to 187 there. So what this is telling you is where those exon-exon junctions are in the sequence. So we've got that information. We also have the CDS, which stands for coding sequence. What this is telling you is that in this whole transcript, the amino acids, or the codons that encode the amino acids, are from base 16 to 618. So bases 1 through 15 would be part of that 5' untranslated region. 618 to 775 would be part of the 3' untranslated region. And within that coding sequence, you have codons for these amino acids. So this would be the amino acid sequence of your protein. And out of those amino acids, you might have signal peptides. Now these signal peptides are going to usually be on the N or the C terminus, and within the cell, they're going to direct that protein to a specific compartment or location. So for example, this IFNL1, interferon lambda 1, is a cytokine that is secreted from the cell. So this signal peptide that goes from position 16 to 72, that is a protein sequence up here on the N terminus that would signal for this protein to leave the cell, to be exported somehow. So it's important that we know that, because oftentimes the mature protein lacks this signal peptide. It gets cleaved as the protein is secreted or directed to a specific location. So while this is the original amino acid sequence of the protein, what this is telling you is that it's probably only this sequence that is in the mature peptide. Okay, so there we go. And we can also scroll all the way down to the bottom and get the entire transcript sequence. So all those numbers that were up above, we can see where those come in right now. So for example, the first exon goes from bases 1 to 186. So that would be from here to about there. That's your first exon, and then this would be your second exon that comes after it. Okay, so what I'd like you to do is copy that sequence and then let's go back to the PowerPoint presentation. So you can see everything that I've talked about so far is in here. Let's go to this website next. Now, this is strictly optional, but I really like the convenience of this. You can paste that sequence into this window here, then remove all the numbers and spaces from it. Get rid of that word origin as well. And that makes this sequence a lot easier to search later on. So if I just open Word here, there we go. I've got my sequence. I always change that to Courier New for the font. That makes every letter the same size. It's just a lot easier to work with that way. But there we go. This is my interferon lambda 1 transcript. And if I go back to the entree entry, I can start to annotate it. So that coding sequence goes from 16 to 618. If I come back in here to my Word document, I can find base 16. There we go. As you can see down here, that is the 16th base. And sure enough, there's an ATG for my start codon. So this over here would be then my 5' UTR. Then I have my coding sequence that goes on here until base 618. So let's figure out where that is. Base 618 right here, and sure enough, that is one of the 3 stop codons. Stop codon can be TAA, TGA, or TAG. So there you go. Everything in here is my gene. So those are all the amino acids that code for my protein, or all the codons that code for my amino acids. And then down here would be the 3' UTR. We're not done yet though. Remember, we have exons as well. So from base 1 to 186 is our first exon. Okay, so we would actually have an exon junction right here. Let's see, where's the next one? It would be base 264. All right, you can see that a lot of these exon junctions are GG motifs. That's very common in intron splicing, but not necessary. All right, so there you go. Now I know where my exons are. Let's see, for IFN lambda, there was also that signal peptide from 16 to 72. So let's mark that as well. All right, and the colors I'm using here are arbitrary. You could make them anything you want, but I'm going to put a gray color there to mark the signal peptide. All right, so I didn't mark all the exons here, but you can get the idea of what this gives us now. We have a very clear picture of the gene, and we know where we should cut with Cas9 to make a loss of function mutation. So very importantly here, I would not want to cut in either of the UTRs. You can see there, if I make a mutation in the UTR, it's not going to affect any of the amino acids in the protein. Likewise, I'd also prefer not to mutate the signal peptide here either. Now, if you made the right mutation, maybe you could prevent this protein from being secreted from the cell, but it would still be expressed. Instead, I would like to have a different type of mutation somewhere here in the mature peptide that completely disrupted the protein or prevented it from functioning properly. So ideally, I would want to target somewhere, let's say in this first exon, after that signal peptide. And by the way, it's a lot better to make a mutation early on in the gene, somewhere up here in the first exon, as opposed to the last few exons, because early mutations will interfere with the way the protein is folding as it's being translated. This is where the protein starts to be translated. So if you can make a mutation earlier on, you have a higher chance of destabilizing that protein to where you won't see it anymore. All right, so there's the transcript. Now, let's go back to the slides. Okay, so that's what the fully annotated sequence would look like. And next, let's look for the promoter for this gene. Now that Entree entry did not have the promoter sequence. It only contained the mRNA sequence. So to get the promoter as it exists in the genome, we'll have to use a different tool for that. In this case, I'm going to show you how to use this eukaryotic promoter database. So I'll click on that, bring up that website. There we go. Just like Entree, what we need to do is type in the gene ID. All right, so interferon lambda 1, and we're going to search for the human version of that. Okay, and that brings us right to this entry here. We see all the usual information. So this is IFNL1, it comes from human. Right here, this is not the sequence of the promoter. Instead, what this is showing you is this is the start of the transcription from the genome, and then everything upstream of that is where the promoter might be. Okay, so capital letters would be the bases that are transcribed. These are not transcribed. That's where the promoter is. Okay, and then down here we have some ChIP-seq data. So ChIP-seq data we could talk about for a few hours, but essentially what this is showing you is where proteins are bound to different regions of the sequence. So for example, we need RNA polymerase to transcribe this gene. And what this blue diagram here is showing you is where RNA polymerase 2 binds to this promoter. So you see a few different peaks here. So that's good. That highly suggests that the promoter sequence for this gene is in this area right here. And RNA polymerase probably binds to one of these peaks and then starts transcribing. Above it, in red and green, these are some histone modifications. So for example, histones are the proteins in the nucleus that bind DNA and condense it. And histones have several subunits, and each one of those subunits can be modified post-translationally. So for example, this is showing you the H3 histone subunit has a lysine, that's the K, at position 4, and it gets monomethylated. So there's a methyl group that gets added onto that lysine. And this H3K4ME1, likewise H3K4ME3, which is just the trimethylated version of this modification, both of these marks are commonly associated with histones that are in promoters. So if we see a lot of peaks here, that means that we probably have a promoter region. This is an area of active transcription, so it's good to see these peaks there. And the position of the peaks tells you where those histones would be. Okay, so this looks good. And then we go down to the meat of the matter, the sequence retrieval tool. So this is where we can actually retrieve that promoter sequence. Instead of sifting through 6 billion bases of DNA, I can do that automatically here. So all we need to do is enter a window here that we want to retrieve. And this window is relative to the transcription start site. So if I go back to my sequence that I just obtained for the transcription start site is right here. It's this A. And we saw that as well up here in the promoter sequence. You see it starts AGTT. We see the same thing here, AGTT. Okay? So within the transcript, we have 15 bases here that are upstream of that ATG. Oops. So if I'm looking to get 1,500 bases upstream from the ATG, then I would put in 15 right there. Because you have the transcription start site at that A, then it goes 15 more bases, and you get to ATG. And then if I want 1,500 bases total, what I would put in here would be negative 1485, okay? Because 1485 plus 15 gives me that 1500 total bases that I want upstream of that ATG. All right. But since we already have the 15 bases in the transcript, I'm just going to put a 0 right here, and that'll make it easier to append to that transcript later. All right, so let's get the sequence, and here we go. This is the sequence of your promoter. Well, it could be the sequence of your promoter. Genetics is a complicated and funny thing. Usually, RNA polymerase binds in this area right here, and you have transcription factors that control RNA polymerase binding in this area, but it's important to mention that the genome is not one-dimensional. It is a three-dimensional object. So we might have transcription factors that control this gene that are tens of thousands of bases away because the DNA can fold to bring those transcription factors closer to this promoter region here. They could even be on another chromosome for all we know. But this is a good first guess that our promoter sequence is right here. And this is everything we need to know about the regulation of this gene. So I'm just going to copy that and then come back into my document right here. And there we go. So that gives you the sequence of your promoter and your transcript. Let's see if I forgot anything, but here we go. That's everything I talked about. And yeah. Oh, one last thing. Once you have this promoter sequence, you can try to find the TATA box as well. So the TATA box is usually where RNA polymerase binds to a promoter and then starts transcribing. Now, it should be around 30 bases upstream of your transcription start site, but it's not always perfect, and sometimes it's completely missing. So for example, I would like to see TAA here, but instead I see T with a bunch of A's. Now this would be a relatively weak TATA box, but it is still there. Some promoters don't even have a TATA box, so don't worry if you don't find one. That might be the case, but your gene still gets transcribed. Okay. So lastly, let's see what else we can learn about this promoter. Now for that, we're going to go to this promo website right here, and we're going to search the promoter for transcription factor binding sites. So let's open up that website. Okay, good. This is what promo looks like. Pretty simple tool to use once you know how to use it. First of all, we're going to select species. And we are looking at a promoter that's in a human cell, so we only want to consider human factors and human sites. Let's click Submit there. Then we want to click on Search Sites, and this is where we input the promoter sequence, and we hit Submit. Now this may take a few seconds, but what this will give you is a list of the different transcription factors that might be binding to your promoter. So you can see there's usually a lot, especially if we're giving this program 1,500 bases of sequence to search through. It's going to come up with a lot of these different transcription factors. So for example, here's NFKB. So this is not surprising to find NFKB in this promoter sequence because NFKB is part of the innate immune response. So whenever the cell is infected, NFKB will be activated. And one of the things that we know NFKB turns on is cytokines like interferon lambda. So it's not surprising to find an NFKB site here in the interferon lambda promoter. We can click on any one of these entries to get a little bit more information about the transcription factor. Specifically, here's its consensus sequence. So it likes to bind that sequence best, but just like with the TATA box, you might see some errors when you're looking for this in the actual promoter sequence. One or two of the bases might be a little bit different, but as long as you see most of them, it's highly likely that NFKB actually does bind there. Okay, so we've got a long list of transcription factors. You don't need to write down all of these. Just be familiar with this and maybe save this web page for future reference. And then down here is where all those transcription factors bind. So you can see these numbers up here are the base numbers for that sequence that we uploaded. We uploaded 1,500 bases, so you can see down here, this would be the end of it. And if you look at this, it's pretty interesting to note that there's a lot of these factors that bind pretty close to the end of the promoter, which is also the transcription start site. So that should make sense because RNA polymerase is binding down here. So there's probably a lot of other transcription factors that help it to bind there. And by the way, we're not absolutely sure that all of these are binding to a given location. It's probably just one or two of them in a given space because a lot of these sites overlap. And depending on the conditions in the cell, 14, for example, might be present at one time, 55 is present at another time, and that determines which one binds. Okay, so that's everything that I had in this slide deck. But there you go. Now you've got the sequence of both your gene and your promoter, and we know a lot of things about it. We know where the exons and introns are. We know what transcription factors are in the promoter, are binding the promoter. So now we can move on to the next steps in our experiments.